Welcome back to the next part, which is about non-additivity in polyploids. So indeed, polyploids can lead to non-additivity of the traits. So, what do I mean by non-additivity? Consider here parents, parent 1, parent 2, and when we talk about additivity or non-additivity, it's always a comparison of the progeny of these parents, let's say, compared to the parents. And imagine that you have a trait, parent 1 has a higher value for this trait compared to parent 2. We talk about additivity when the value of the progeny is equal to the mid-parent value, and the mid-parent value is just the average between parent 1 and parent 2. So basically it's Mendelian expectation that you have, which is parent 1 is this, parent 2 is this, and then you have here additivity and no case of dominance or anything. Now imagine that the progeny actually has a value which is closer to parent 2 here and parent 1 here or here, doesn't matter. Then we will talk about dominance, and in the case of allopolyploids we will talk about homologous dominance or homologous bias, which means again that the allotetraploid look more like one of the parent or the other. The last case that is non-additivity is when the progeny will have a higher value compared to all the parents or will have a lower value compared to both parents. And in this case we talk about transgressivity. And this scenario can happen in polyploids, and we're gonna explore why and how. But first let's take an example. Here we are in Nicotiana, so tobacco basically, and you have two parental species, diploids, both of them, same number of chromosomes, and they are the origin of allotetraploids here that originated around 1 million years ago, okay? And then the author created the same basically allotetraploid, but synthetic, which means they crossed these two species and made a tetraploid, and this is like 2 years ago, so you can say 0. And then what this author did, they compared the floral morphology, so the morphology of the flower, in the parents versus the allotetraploids originating from these parents, both natural and synthetic. So how does it work? According to the shape, basically you have coordinates of the species, and you have here one of the parental species in blue and the other is in red here. And then what you have here in the middle is the mid-parent value, and the expectation in case of additivity is that the allotetraploids will look like or will have a similar shape as the mid-parent value, okay? And that is represented by this line that you have here. Those lines are actually the distance between the mid-parent value and the actual value of the allotetraploids. And then they studied natural allotetraploids, so Clevelandia, one that I showed to you, but there are others from the same parents, and the synthetics allotetraploids. So let's look first at the natural ones. They are here. As you can see, they are quite transgressive. They don't look like any of the parents, or maybe let's say they look a bit more than these parents, but still has a different value compared to all of them. And the distance from the mid-parent value is very high. So here we can talk about transgressive phenotypes. Of course, one problem here is that those allotetraploids diverged from the parental species quite a long time ago. So they had time to evolve differently, and that could be just due to this evolutionary time and nothing else. So that's why we should instead look at the synthetic allotetraploids that are here, but those didn't have time to evolve, or almost no time. And if we look first at the pink one of the lines, you can see that, okay, we can consider this as mid-parent value. It's still in the range of it. You see that the distance is pretty low, and the pink cloud, which is variability within this allotetraploid, is within the gray cloud, which is expected as mid-parent value. But now, if we look at the blue here, or the green here, especially the blue one, you can see that actually it diverges quite significantly from the mid-parent value. So it gives a transgressive morphology of the flowers. So again, the same pattern seems here, that within a very short time you already see a difference from the same formula, the same allotetraploidy origin. I mean by that 3 different synthetic allotetraploids from the same parent already show signs of divergence between them. That could be due to the genomic instabilities that we discussed before. And the other thing is, indeed, you see that you have non-additive phenotype of those allotetraploids. Okay, so what are the causes for this non-additivity in polyploids? Well, first, let's consider a tetraploid. One obvious thing is that with more chromosomes, you can have more alleles. So in diploids, you can only have 2 alleles, and you can be homozygous for one or the other, or you can be heterozygous. But in the case of a tetraploid, you can get up to 4 different alleles. You can of course be homozygous for one, but you can go up to 4 different alleles within the same. And of course, this allele combination doesn't exist in a diploid, so that can lead to transgressive phenotype. And another point is, instead of having different alleles, you can consider that allele have a certain dosage, and then you can have a different combination of those dosage. Let's consider here, for example, like a diploid, so heterozygous parents, which leads to the traditional Mendelian segregation. But in the case of the tetraploid, you will have different dosage between these 2 alleles happening. So of course you can have again homozygous for one or the other, heterozygous, which looks like the heterozygosity of diploid, but then you can have a dosage of 1:3 or 3:1, which can't exist in the diploid. And that can lead again to some transgressivity. Now, let's go back to the trichopogon example. So remember, so you have these 2 different species that leads to allotetraploid trichopogon species. This is a karyotype of the 2 species, and this is, for example, the karyotype of one of the allotetraploids. So as you probably remember, there is a case of compensated aneuploidy, which means that in one chromosome of one subgenome you have, for example, 4 copies here, but in the whole subgenome you have no copy at all, which means overall you have 4 copies of this chromosome, but only coming from one subgenome. And then, of course, can lead to either transgressivity or homologous bias. And in this case, you expect actually homologous bias, and you expect that this subgenome will actually drive the trait of this allopolyploid, and this allopolyploid will look more like this species, let's say. And the last case, I won't go further because evidence are still missing, but the last proposed hypothesis is that epigenetics could play a role in non-additive phenotype of polyploids. So epigenetics is the regulation of genes through chromatin modifications that can be inherited through mitosis or meiosis. And then one theory that has been proposed is that instead of having a lack of some chromosomes or some new genetic dosage or genetic composition of the genome, you could have just expression patterns that are different between chromosomes. Let's say one chromosome could be silenced, the other one could be fully expressed, and that will lead to some non-additive phenotype. So the authors here check this hypothesis. So you have a diploid versus a tetraploid. They stain the chromosome in blue with just DAPI, so it just stained DNA. And then one of the marks that contributes to epigenetic silencing is DNA methylation. And again, here it's in situ hybridization with a probe having a fluorescent molecule, which is here pink. And let's look at the diploid first. So in the diploid, you see that the pink is basically widespread in all chromosomes in a very homogeneous manner. You have more or less the same amount of DNA methylation in all chromosomes. But now, if we look at the allopolyploid, so this is autopolyploid, you see that here, for example, this chromosome lack mostly DNA methylation, but this one here, for example, has very high enrichment in DNA methylation. That could suggest that what's happening is that in this chromosome, you have expression of genes to a high level, but this one here is fully silenced. And that could lead, of course, to some transgressivity, unless it's just a dosage compensation, again, with this one being expressed, compensating with this one. Of course, this is just a preliminary study, and more is need to be known in terms of genetic expression, but that's a possible mechanism that needs more exploration in the future. Okay, so as a conclusion and as a little quiz, as usual, I ask you to tell me what is non-additivity. Then I want you to give me, with a maximum of 6 words for each, 3 reasons of non-additivity in polyploids. It can be what we discussed, or if you have other ideas, feel free to tell them. Finally, here is a karyotype of one of the Brachopodon allopolyploid individuals, and I would like you to tell me what is your expectation in terms of additivity compared to the parental species. If you look at this karyotype, do you think you will have additivity or not? And if not, is it like more transgressivity, homologous, dominance? Good luck.